Yes, this is microbiology. Today we'll be discussing somewhat about the basic classification of bacteria. So here is the basic classification of bacteria is based on the gram staining, different fermentation, and also the classification based on the antibiotic sensitivity and resistance, and also on a base of different biochemical testing. So there is a main and important flow chart of the bacterial classification. So let's start first the gram-positive bacteria. So on a gram stain, gram-positive bacteria will be seen purple or blue in color, while gram-negative bacteria we can see under microscope on a gram stain. It will appear pink or red in color. So gram-positive bacteria further divided into two on the basis of their shape. First is cocci, gram-positive cocci, and the second one is gram-negative rod or bacilli. So the gram-positive cocci data in round in shape, uh, while gram-positive rod are in rod in shape, or bacillus. Also called bacillus, they are in rod shape. So first discuss about the gram-positive bacilli or rod shape. It contains a bacterium Clostridium, Corynebacterium, and Listeria bacillus. And the second one is the gram-positive cocci. Gram-positive cocci further divided into two subgroups that are Staphylococci, gram-positive Staphylococci, and gram-positive Streptococci. First talk about the shape of these two bacteria. Staphylococci are in cluster palm, and while Streptococcus they are in chain palm. And the other difference between these two bacteria are that the Staphylococci are catalase positive, while the Streptococcus are catalase negative. So further, Staphylococcus has other species like Staphylococcus aureus, Staphylococcus epidermidis, Staphylococcus saprophyticus. So the Staphylococcus species are further differentiated between each other. That are Staphylococcus are coagulase positive, while Staphylococcus epidermidis and saprophyticus are coagulase negative bacteria. So further, the epidermidis and saprophyticus differentiate on the basis of antibiotic sensitivity and resistance. So Staphylococcus epidermidis are neobiocin sensitive. Neobiocin actually is antibiotic. So epidermidis are sensitive to neobiocin, while saprophyticus are resistant to neobiocin. So let's talk about the gram-positive streptococci. So streptococcus species are further classified on the basis of hemolysis. So the first one is partial hemolysis, is also called alpha hemolysis. It contains two bacterial species that are Streptococcus pneumonia, while the second one is this Viridon group. It contains Streptococcus mutans and Streptococcus sanguis. So the Streptococcus pneumonia are capsulated, and also they are sensitive to optogene antibiotic, while the Streptococcus viridon group they have no capsule, while they are resistant to optogene antibiotic. So the second one is the Streptococcus beta hemolytic streptococci. This contain two group. A group it contains Streptococcus pyogenes. So Streptococcus pyogenes that are bacitracin sensitive. They are sensitive to bacitracin antibiotic. While this group B it contains Streptococcus agalactiaceae. They are resistant to bacitracin antibiotic. So why they are called partial hemolysis? Alpha hemolysis is also called partial hemolysis because the group of this bacteria is partially hemolyzed the blood cell in a blood culture, while beta hemolysis are called clear hemolysis or complete hemolysis. It clear it it broken down the all blood cells in a blood area, so that is why it's called clear hemolysis. So the third one is the no hemolysis, or it's also called gamma hemolysis. So it contains a species that is Enterococci, E. peccalis, E. fecum, and Peptostreptococcus. So these species are not involved in any hemolysis. So the group of partial hemolytic bacteria have their green color appear around their colonies, and while in the beta hemolytic bacteria, it may be A or B group, have their yellow color appear in their colonies, while in gamma hemolytic bacteria species have no any color found around their colonies. Gram-negative bacteria. Gram-negative are also further divided into two: gram-negative cocci and gram-negative rods. So the gram-negative, when we perform gram staining of this bacteria, it will appear pink in color, or it may appear in red in color. So the gram-negative cocci are further divided into two species. There are Neisseria meningitis and Neisseria gonorrhea. So the further these two species are differentiated on the basis of maltose fermentation. So the Neisseria meningitis, they are maltose fermenter, while Neisseria gonorrhea are non-maltose fermenter. And the second one is the gram-negative rods, that is called GNR. Gram-negative rod, they are further divided into two. They are on the basis of lactose fermentation. One is non-lactose fermenter, and the second one is lactose fermenter. So the lactose fermenter further divided into two group. That may be slow fermenters. So the slow fermenter bacteria are Citrobacter, Serratia species are others, and fast fermenter include E. coli, Klebsiella, Enterobacter. So these bacteria are lactose fermenter, and they are further divided into two groups. They are slow fermenter bacteria, and also it contains a fast fermenter bacteria. So the fast fermenter bacteria is E. coli, Klebsiella, Enterobacter, while slow fermenter bacteria is the Citrobacter, Serratia, and other groups. So the non-lactose fermenter bacteria are also further divided into two classification. So they contain two group of bacteria. One is oxidase positive bacteria, and the second one oxidase negative bacteria. So this is actually oxidase is biochemical test. So here we classify the group on the basis of biochemical test, that is oxidase test. So oxidase positive bacteria are Pseudomonas, Plasmonas, and oxidase negative bacteria is Shigella, Salmonella, and Proteus. So that's all about the basic classification of bacteria. I hope you will be understand. Thank you for watching. The third one, the remaining last one is the gram-negative, contain cocci rod bacteria. It may be in round in shape, it may be found in round shape or in a rod shape. So the first one is Coccobacilli or Haemophilus influenza. It required factor V and X. It's basically is a growth factor that is required for this Haemophilus influenza. And Pasteurella, uh, Brucella, Bordetella, pertussis. These are those bacteria. They have it may be found in cocci or in a rod shape. These are the group of bacteria that may be found in both shape, either cocci or bacilli.